Hello everyone, this is Dr. K. Geeta from Department of Biotechnology, Kamaraj College of Engineering and Technology, Virudunagar. Today we are here to discuss about how antigens are processed and presented for them to be recognized by a T cell and processed. So let us see some background related to that. So as you know, for an antigen, when it comes into the body, for it to be recognized by the T cells, it requires the MHC molecules to present them, and this is known as MHC restriction. And as for the antigens, we have two broad classes of antigens, as you know, the endogenous antigens and the exogenous antigens. These endogenous antigens are presented or associated with class 1 MHCs, whereas the exogenous antigens are associated with class 2 MHC. To put it in more simpler words, endogenous antigens are presented by class 1 MHC molecules and exogenous antigens are presented by class 2 molecules. Let us understand how this is happening. But before we delve into that, let us see a brief overview of what these MHC molecules are. So as you know, MHC or major histocompatibility complex molecules, they are broadly of three classes, and out of which class 1 and class 2 play the major role. MHC class 1 can be found on most of the nucleated cells, hence they can be, or the antigens which are presented by class 1 MHC molecules can be presented by almost all the cells to the T cells, whereas MHC class 2 molecules, they can be presented by only the antigen presenting cells, as they are expressed only by the APCs or antigen presenting cells. And as you know, antigen presenting cells are of two categories. We have professional antigen presenting cells, where we have three types of antigen presenting cells called the dendritic cells, B cells and the macrophages, whereas when we talk about the MHC class 1 cells or the cells expressing MHC class 1, all the nucleated cells have the ability to express MHC class 1. So coming to the types of T cell which are involved with different types of antigens, we know there are two major types of T cells called the CD8 cells or the cytotoxic T cells, and the CD4 cells or the T helper cells. And CD8 cells are presented with an antigen by class 1 MHC molecules, and the antigens that are presented are the endogenous antigens. And on the other hand, the T helper cells, they are presented antigen by class 2 MHC molecules, and the antigens which are presented are the exogenous antigens. For example, the antigens presented or the antigens related to the bacteria. So antigen processing overall can be classified into two pathways. The first one being cytosolic pathway and the endocytic pathway. These two pathways are categorized based on the type of antigen involved. So if it is endogenous antigen, we call that as a cytosolic pathway, and if it is an exogenous antigen, we call that as the endocytic pathway. So here the type of antigen involved will differ and the type of MHC molecules involved will differ. So at a particular point, you will see both the pathways, whether it is cytosolic pathway or it is endocytic pathway, both of them merge together to almost the same kind of ending. So in case of cytosolic pathway, the protein or any antigen which comes in, that is broken down by the cytoplasmic proteasome complex and converted to the peptides, which are then transferred to the endoplasmic reticulum, from where they will associate themselves with class 1 MHC molecule and present it on the surface of almost all the nucleated cells. On the other hand, in case of endocytic pathway, majority of the processing happens via either endocytosis or via phagocytosis. And once the processing happens, this is then followed by, again, within the cell, whichever cell is involved in endocytosis or in phagocytosis, the protein or the antigen which is taken in is then broken down to peptides, and again, just the way we saw in the cytosolic pathway, here also the peptides will then associate themselves with class 2 MHC molecules in this case and presented on the surface of the antigen presenting cells. either professional or non-professional. There will also be a scenario where these peptides may also be acted upon by exopeptidases, wherein they are broken down to amino acids which are recycled. So, this is the overview. So, let us understand each of the pathway in little more detail. The first pathway is the cytosolic pathway. Overall, the entire cytosolic pathway can be divided into 3 steps. The first step being peptide generation from the process of proteolysis. So, in this case, when a antigen comes in this case we are just taking the example of a peptide, it is then complexed with ubiquitinating enzyme called the ubiquitin, and this complex then is processed by the proteolytic enzyme subunit called the proteasome. The proteasome processes the ubiquitinated protein to produce peptides. In the next step, you will see the peptides are then transported with the help of TAP, which is an abbreviation for transporter protein associated with antigen processing. This TAP is basically a heterodimer which uses ATP and transports peptides approximately 8 to 10 amino acid long to the lumen of endoplasmic reticulum. Once it reaches the lumen of endoplasmic reticulum, there the peptide will bind with the MHC class 1, and for the peptide to bind to MHC class 1, there is a small processing that needs to happen, wherein multiple proteins are involved, like you have the class 1 MHC alpha chain which combines with calnexin, and this results in formation of calnexin associated class 1 MHC alpha chain, and this complex then associate itself with beta 2 microglobulin, resulting in formation of another complex which also has tapacin and calreticulin involved. So, this entire complex, when it comes in contact with a peptide, it can be any kind of a peptide, in this case our antigen. This will lead to dissociation of calnexin, tapacin and calreticulin, finally giving you the class 1 MHC molecule associated with our protein. And this then exits the endoplasmic reticulum and is then presented on the surface of the cell. So, this is the overall picture of cytosolic pathway. Then comes endocytic pathway. As I have pointed out, endocytic pathway is involved in processing of exogenous antigens, and here again it is either through endocytosis or through phagocytosis. So, there are the pathway totally involves few stages like formation of early endosomes, then late endosomes which are also called as endolysosomes, where you have the lysosomal proteins involved, and finally lysosomes where the actual degradation of the protein happens. So, here is a pictorial representation of the entire process. You will see when an antigen comes in contact with a receptor on the surface of a antigen presenting cell like a B cell, it is taken in, and there you have formation of clathrin coated vesicles. These vesicles then further are converted to endosomes, late endosomes, and finally lysosome, where in presence of lysozyme the peptides are further, the protein is further broken down into peptides, and then they associate with MHC class 2 molecule, which are then transferred to the plasma membrane and then presented on the cell surface. So, here just like the cytosolic pathway, again the class 2 MHC manufacturing requires few stages, wherein the alpha and beta chain of class 2 MHC interacts with a protein called invariant chain, which is actually CD74, and this chain has the ability to go and bind to the protein or peptide binding cleft, and once it goes and binds, in presence of another protein, there will be degradation of this invariant chain, leaving behind a small portion called as CLIP. CLIP stands for class 2 associated invariant chain peptide, and this CLIP, once you have the formation of this complex, if this comes in contact with a peptide, the peptide will come and replace the CLIP in presence of HLA-DM and HLA-DO, and because of this you have formation of MHC class 2 peptide complex, which is then transferred onto the surface of the plasma membrane and thus presented to T helper cells. So, this is the overall picture of the two pathways. This is your endogenous pathway, and this is called as, which is also known as cytosolic pathway, and the other one is the exogenous pathway or endocytic pathway. So, this way whatever antigen is coming in, when it comes in contact with either MHC class 1 or MHC class 2, they are first processed and then presented on their surface, on the surface of the cell, to be taken up, recognized and taken up by the T cells and then further processed. So, thank you very much for listening. I hope you have understood the concept of antigen processing and presentation. Thank you.